Good morning. Yes, now it works. Good morning and welcome to the first session of the conference, which is the acute pancreatitis one. We have the announcement for a move, actually, so we have a change in the last speaker. The last speaker, Attila Ibert, was moved to the session 3, and instead we have another presentation by Felix Juhas, and so I will announce his title later on. And I would like to start our session with the first speaker, who is going to be Zoltán Rakonczay. So Zoltán will present and introduce the session, discoveries by Hungarian researchers in the field of acute pancreatitis, basic research overview.
Thank you, Julian. Thank you for the opportunity to present the basic science part of the HPSG activity. We only have about 15 minutes. And of course, I would have to select the topics from the topics. But I would like to first start with an introduction. So acute pancreatitis, I'm sure, as many of you know, are mainly caused by alcohol consumption as well as biliary disease. So these etiological factors like ethanol and bile acids are also important with respect to the basic science part that we were involved in. The pointer doesn't seem to be working again. Okay, great, thank you. So the classification of the disease ranges from mild to severe. And in the severe form, there's a huge mortality, which is at least partly due to the lack of knowledge that we have in case of pathogenesis. Our main focus in this talk is going to be the investigation of the pancreatic duct cells in acute pancreatitis. So to understand what I will be talking about, I first have to briefly introduce the role of pancreatic ductal cells, which is going to be pancreatic bicarbonate secretion. And without getting too much into the details of this, I would like to highlight two of these transporters, well, actually even three, that I'll be talking about. This is the cystic fibrosis transmembrane conductance regulator, which is a chloride channel, but it can also conduct bicarbonate. And it also affects the activity of other transporters like SLC26A6 transporter, which is a chloride bicarbonate exchanger. And these transporters are mainly responsible for secreting the huge amounts of bicarbonate, up to 140 millimolar bicarbonate into the ductal lumen. And this will also, of course, drive fluid secretion because with ions also comes fluid. So fluid movement is partly regulated by Aquaporin channels, and I'll be highlighting Aquaporin 1 in this lecture. So our aim in these studies that I'm going to be highlighting to you is to determine the role of pancreatic duct cells in acute pancreatitis. And I will be mainly focusing on research work that appeared in 2012, from 2012. This is an exception, so this is before counting. To understand the role of pancreatic duct cells, we also have to mention bile acids, the effects of bile acids. And it's also similar to what we see in case of ethanol. In smaller doses, smaller concentration, the effect of bile acids is going to be a stimulation, which is mediated by calcium signaling, which produces a oscillatory calcium signaling. And whereas when you have a pathological calcium signaling, this will give rise to decreased bicarbonate secretion by these cells. This is largely mediated by the destruction of the mitochondria. This is sort of similar what we also see in S inertia. You have a secretory block in these cells, which will contribute, obviously, give rise to acute pancreatitis. But the important thing is just not only acinar cells, but also the role of duct cells were highlighted by this study, which was one of our first studies by Victoria Banglavets. So our first gut article as well. I think that a huge work that was also took many years to complete was performed by József Malét. And what he's found is that if you have a massive alcohol consumption, that will actually increase the sweat chloride level. And this means that there's an inhibition of the chloride channel activity. And if you take a look at the expression of the cfdr chloride channel, you see that in the membrane of duct cells, physiologically, you have, yes, thank you, this helps a bit. You have expression in the luminal membrane. In case of acute pancreatitis, there's a marked reduction of chloride, cfdr expression. This was also carried out in case of in vitro experiments. So if you check the ethanol and its metabolites, various fatty acids like palmitoleic acid ethyl ester or palmitoleic acid, it can also affect the bicarbonate secretion. So if you take a look at the swelling of these ducts in response to various compounds, ethanol versus the control, here's the control, ethanol dramatically decreases the swelling of these ducts, which means that the fluid secretion by these ducts are markedly reduced. And this is similar in case of palmitoleic acid as well. If you take a look at patch clamp experiments, so you can see that results in that various doses of ethanol or palmitoleic acid markedly reduce the chloride current in these cells. This is also confirmed by the ammonium pulse technique. If you look at the recovery from alkali load, you see that ethanol markedly reduces base flux in these cells. There's also an elegant set of experiments carried out by Linda Junak, who's our PhD student back at that time. And you can see that Mr. ethanol, there's an inhibition of chloride channel, cfdr. And if you administer ATP to these cells via patch clamp technique, then you increase the ATP activity inside the cell and you cause the activation of the chloride channel. So it seems like these common factors, bile acids, ethanol, and its metabolites have an effect both on ductal cells and acinar cells, which cause mitochondrial damage, cause ATP depletion, and impaired cellular function. To determine the role of ductal cells, we also had a look at some NHRF1 knockout animals. NHRF is an adaptive protein that keeps the cfdr protein in the apical membrane. So as you can see that in case of NHRF1 knockout mice, there's a marked reduction of cfdr expression in the apical membrane. What this actually causes, if you stimulate the pancreatic ducts in vitro with forskolin, which is a CAMP agonist, which is a regulator for cfdr channel, there's an increased bicarbonate secretion in these animals that have the NHRF, and there's a reduced bicarbonate secretion in knockout animals. In vivo experiments were also carried out. So secretin is a main stimulant for pancreatic duct cells. As you can see, in wild type animals, there's a marked increase in bicarbonate and fluid secretion in this case. And if you knock out this NHRF1, there's a reduced basal secretion and there's a reduced stimulated secretion as well in these cells. So we're also wondering what the acute pancreatitis will be like in the animals that have reduced cfdr expression. And as you can see, that if you administer large doses of cereulin, which induces a necrotizing pancreatitis in these mice, you get an increased damage of pancreatic acinar cells, especially in the knockout animals, which are lacking cfdr as well. This also shows the very important effect of CFDR expression in these duct cells. Carrying on further, just to exclude some non-specific effects, we've also tested the effect of Aquaporin 1 deletion in animals, which will also result in a markedly reduced fluid secretion by the ducts. This was confirmed by in vitro experiments in the pancreatic ducts, and also MRCP measurements in vivo animals. And very interesting that, that the chloride bicarbonate exchange activity, which is also regulated by CFTR, is also affected in this case. And there's a markedly reduced chloride bicarbonate exchange activity in the knockout animals as well. We've also tested the severity of acute pancreatitis in these animals. And as you can see, that administration of cereulin induces pancreatic damage. And this pancreatic damage was markedly larger in case of Aquaporin 1 knockout animals. Of course, also the question arises how acute pancreatitis itself may affect CFTR expression. So we've also tested how CFTR expression is changed during the course of acute pancreatitis. CK19 is just a ductal marker. And as you can see that in control animals, there's a nice structure that we can see in these animals, whereas in case of acute pancreatitis animals in 24 and 48 hours, there's disturbed expression. This nice structure is completely disorganized. And we've also had a look at various drugs that act on CFDR, VX661 corrector, and the potentiator VX770. And when we administered this during the course of acute pancreatitis, starting as a pre-treatment, we found that both at 24 hours and 48 hours, there is a reduced severity of acute pancreatitis in these animals. So overall, our conclusions drawn from these experiments is that alcohol and acute pancreatitis itself will disrupt the expression and the localization of CFTR. This is probably also true for bile acids as well. If we knock out Aquaporin 1, this reduces the pancreatic fluid secretion, and the pancreas will have an increased susceptibility to pancreatic inflammation. We've also shown that treatment of acute pancreatic mice with potentiators and significantly reduced the extent of pancreatic damage. And overall, we can say that ductal secretion has an essential role in protecting the pancreas from this dreadful disease, and also that ductal function should be an important modality in the prevention and treatment of the disease. So I have to say that, again, I had only 20 minutes to present my talk, and I didn't have a chance to talk about various treatment alternatives, which also we have numerous collaborations with Anna Kukovskaya. There's a collaboration with a national partner, the University of Debrecen, and also with the group of Julia Mayele in Germany. And there's another publication that just appeared in the middle of last year from, again, Anna Gukovska's lab. The first authors were also coming from the University of Szeged in this case. So I'm pretty sure that this list of acknowledgements is not complete. I'm sorry about that, especially the youngsters. Probably there should be a third line. I don't know every bits and pieces, especially in the group of Page, just recently developing group in the University of Semmelweis. But I hope that this list will actually grow and grow. I think the numbers, even if we would stick to this, are quite impressive. And of course, I would like to highlight the role of Peter Hegyi, the main catalyzer of this initiation. And also, I would like to acknowledge our international partners from mainly from the University of California, actually with Miklos moving to the University of California. This made it more easier. We usually have the collaborating partners in Boston, but now it's mainly UCLA. But anyway, so I've mentioned Julia. We also have connections with Marcus and with the NHRF mice. We had this collaboration with Ursula Zeidler. And Peter mentioned that we started out, both Peter and I started our doctoral research in Newcastle. So our mentor there was Michael Gray. I hope that he will be able to make it. And in Hungary, we have some very close connections with Gabor Varga as well. So with this, I would like to thank you for your attention. I don't know whether there's time for questions or Peter. Yes, thank you.
Yeah, thank you very much. It was a beautiful talk. And yes, we're open for questions.
Conclusion about the importance, of course, of the duct fluid secretion. There is also, of course, a fluid secretion from the acinar cells. And I wonder whether the Aquaporin knockouts, whether you look at the fluid secretion produced by acinar stimuli versus the ones fluid secretion evoked by secreting. I couldn't quite read all the details on the slides.
Yes.
Could you comment on that, please?
Yes. Thank you for the question. Obviously, this is a relevant question, and this was a general knockout mice, so it was not specific to the ductal cells, but we didn't look at the acinar cells in this respect. But the contribution of fluid is much smaller from the acinar cells. But of course, it is possible that the acinar cells also have a role in this process.
Thank you, Zoltán. Quick question about the cfdr correctors effect on pancreatitis. That doesn't really make a whole lot of sense to me. If you knock out cfdr in the mouse, not much happens, right? So why would a cfdr corrector actually improve outcomes?
Yeah, we've tested it and we are actually in the phase of testing the effect. And it is a relevant question, of course, I have to say. The corrector we used because we actually saw that this cfdr expression is markedly disturbed in this, and we also had a figure in the article, Journal of Physiological article. I didn't show it in this respect. And it actually didn't really change the expression of cfdr. So we actually thought that it was mainly the potentiator that had this effect, beneficial effect. And we are currently in the process of testing this as well in the mice. But thank you for the question.
Just one comment to Miklos that unfortunately mice is a terrible model for CFDR and especially mimicking the human issues. So we have two reasons. not supporting this because unfortunately the guinea pigs and the big pigs, which are actually related as a very similar mouse model or animal model for studying the human bicarbonate secretion and cfdr. Unfortunately, we don't have as many as genetic models for guinea pigs.
Thanks. Very interesting talk. And I do believe there's, some merits in this. But my question would be, how do you now perceive to translate this also into kind of human research? Because exactly as Peter was telling, the mouse is not that big of a good model for particular this indication. Do you have any thoughts on how you could expand all these experiments and also into kind of human experiments?
Yeah, but it's quite difficult, and we're always thinking of this. And we had this study, and I didn't show it. I wanted to actually have. There's a study by Tamás Takács in which he measured the intraluminal pH of patients with biliary pancreatitis, and he's found that in case of biliary pancreatitis, with time, there's a dramatic change in pH, which would actually indicate that with time, that the bicarbonate secretion of these patients is going to decrease. Of course, this is related probably not just to the disease itself, but also that the patients are probably not eating, and there's no stimulation of bicarbonate secretion as well. But if your question is related to the therapeutics, it is.
Well, it's more about the physiology, in fact. We are now, for example, taking biopsies from patients with chronic pancreatitis. We go into pancreas when we treat stones, we take biopsies to do some human studies. Also, we now collect with secondary stimulation, we collect juice from the duodenum, and we're now able to culture organoids from ductal cells. Yeah. So I think that's a whole new avenue to interrogate and to investigate this issue, which I think has merit.
I mean, yes. So I'm pretty sure, Josef, are you going to be speaking about organoids as well? So Josef is the main driver of this, and we are also investigating organoids from fetal pancreatic. So that would be much more, it would have more human relevance, although this is in a fetal stage. So we're still investigating how relevant is that to the adult situation. And we are limited to the mid-trimester because of ethical issues, obviously. So we do have some ideas with the organoids that are going to be mentioned by Josef. Also, I have to say that it's much more difficult to isolate ducts from human samples. Josef may be able to comment on this much more because they are investigating this from transplant patients. But the human pancreas is much more fibrotic, especially as you get older. So it's very difficult to obtain these ducts from human samples. Thank you.